Excellent. Good morning. My name is Dan Meyer, CEO of CellChorus, and our CSO, Naveen, did his postdoc work at MIT. We are the dynamic single-cell analysis company. So what that means is that we apply artificial intelligence to watch and evaluate thousands of videos of cell-cell interactions to improve the development and delivery of immunotherapies and other new therapeutics based on their function and performance. Now, as we move into a world where we increasingly rely on cell-based and cell-modifying therapies, there are two really important things to keep in mind. One is that in order to fight disease, cells need to move and interact and perform. And so we need to evaluate the performance of those cells over time. And the second thing is that in any given population of cells, there are important differences. And so it's not sufficient for us to evaluate a large number of cells in bulk, and it's not sufficient for us to evaluate a very small number of cells at single cell resolution. We need high throughput modalities with single cell resolution. And unfortunately, most of our customers, collaborators, and other clients rely on bulk live cell imaging to evaluate the performance of their cells. This means that they essentially get readouts around the target or disease cells across the entire population, and they're not able to determine which of their therapeutic or effector cells are the best performers or why. So instead of placing tens of thousands of cells of different types in the same container and evaluating what happens over time, CellChorus places small numbers of cells in each of thousands of nanowells. And so in this example, you'll see a cell labeled green that's an antigen-specific T cell from a recovered COVID patient. So this cell is supposed to be able to sniff out COVID-infected cells. And then there are 3 cells that are expressing spike protein. And so as we play the video, we see that T cell needs to move, contact, release, and then move again, contact another cell if it's going to kill these cells. And when they die, they appear purple to us based on the labeling in the assay. Now, what that means for our customers and collaborators is that instead of getting average data across the entire population of target cells, they still get average data across the broader population and subpopulation of target cells, but they get data on the effector cells. They get subpopulation data and detailed single cell information about motility, contact, killing, cytokine secretion, et cetera. While we're happy to send all of the videos to customers, typically that's, for a study that's many terabytes of images, but it is important for customers to get example videos of specific subpopulations or biology of interest. So in this respect, when you think about what's happened in life sciences, there have been 2 major trends. One is a move towards single cell analysis that's transformed genomics, transcriptomics, and proteomics. And the other is a move towards multi-omic profiling. And one of the benefits of our platform is that in addition to being the leader in the next wave of evaluating single cell dynamics, we also can select cells of interest for downstream profiling like single cell RNA-seq. So when we work with other groups, we're able to impact the preclinical and clinical development, which is where we spend most of our time right now. And then we also have applications that will launch next year to improve manufacturing and clinical care for cell therapies, antibody therapeutics, and vaccines. And so if you're interested in working with CellChorus, we run a lab in Houston where we receive cells from customers and collaborators and deliver back results. We have done work with companies developing new cell therapies and antibody therapeutics. We, next year, will run our first experiments with vaccine programs. And then on the partnership side, we're interested in speaking with companies about preferred CRO relationships because next year we plan to launch an instrument that will allow the platform to be run in other labs. Also manufacturing both the imaging instrumentation and our nanowell arrays. And then as we move beyond working with North American customers, we're also very interested in speaking with international distributors. If you would like, if you're interested, I can either meet with you in the breakout room or you can find me at dan@cellchorus.com. Thank you.
Thank you, Dan. That was great. So first question, you mentioned the lab in Houston. Could you expand a little bit more? Is it mostly the platform available through the lab, and what does that mean?
Yeah, so today we are operating out of the lab. We receive frozen cells or other reagents, inputs, et cetera, and then we run experiments and deliver back results. Late next year, we expect to place our first instrument with a customer, client, or partner or collaborator.
Great. And how are you working with clients today? What are some things that work for you when you meet with a corporate?
Yeah, so most of the work that we've done today has been prioritizing candidates for clinical studies or IND enabling studies, through to characterizing response on clinical trials. So receiving cells from patients that are responders and non-responders and characterizing the different dynamic and functional performance of those cells to help understand why patients respond or don't respond. And that has proven to be very impactful for customers because ours is the only platform that can correlate on a single cell basis, motility, contact, time to contact and duration of synapse, biomolecule secretion, typically interferon gamma, and then serial killing, mono killing, et cetera. And we have some publications coming out in Q1 that'll be pretty impactful in showing the performance of the platform.